Okay, so this chapter is host microbe relationships and disease processes. Okay, so the first thing they talk about is how the relationship between hosts and parasites or hosts and other organisms. So the first thing we're going to talk about is symbiotic relationships. So symbiotic means they exist together. So usually it's in symbiosis. If you take two things and you put them together, you get something greater than the parts. But they have examples of symbiotic relationships. So there's three different kinds. So the first one is mutualism. So in mutualism, both the host and the pathogen benefit. So both are plus and a plus. So mutualism, an example is termites. So termites actually have bacteria that live in their gut, and they are what gives the termites the ability to digest wood. And humans, you can think about the microbiome and how the microbiome helps us out. Okay, now parasitism is when one, the parasite, benefits while the other, the host, is harmed. So I always think of blood-sucking parasites, so ticks, fleas, worms, things like that. Okay, and then commensalism, this one I think is more challenging because it's hard to find, I think it's hard to find this. So in commensalism, one benefits, okay, and then the other, nothing happens to it, so it's kind of like neutral. So most of the time there's an effect on both, but this is a formal definition that exists. Okay, now when we talk about host invasion and how hosts how the host is invaded, there can be like three kind of stages in it. So we're three different levels. So if something is contaminated, it means the microbes are present. So an example would be a healthcare worker that has Staph aureus on her gloves, or you have bacteria on your pencil. So it's contaminated, but it's not causing any problems. Okay, now infection means that the microbes are present, they're starting to multiply, and they can start to cause disease. So example would be the healthcare worker has a wound and she gets Staph aureus in the wound. Okay, so it's starting to cause an infection. Another term that's used is infestation. So infestation is the term that they use for things like insects or protozoa. So you have like fleas or you have a lice infestation. It's the same idea as infection, but they use it for different organisms. Okay, and then the next step is disease. So disease are when you start to have symptoms or signs. So you start to have something bad happening to the host as a result of the infection. So example would be you get a boil or a sore as a result of having Staph aureus in the wound. Okay, now pathogenicity is the capacity to produce disease. Okay, now if we go down here, virulence is the intensity of the disease produced by the pathogen. So how bad is the pathogenicity is kind of what virulence is. Okay, so now things that can influence pathogenicity are microbial count. So how many microbes are necessary to cause disease? And it depends. It depends on the host. It depends on the disease. It just depends on the environment. Okay, so different diseases have different infectious dose, and they call it ID50. So ID50 is the estimated number of organisms or a virus particle that's required to produce infection in 50% of the normal adult humans that are exposed to it. Okay, so some things have a really low ID50, like norovirus is really low. So norovirus is the diarrhea. So it has a really low ID50, like maybe 10 virus particles. Okay, now virulence. One thing that can happen with virulence is that as a pathogen passes from animal to animal, it becomes more pathogenic. So it gets better at causing infection the more it goes from animal through. If it infects mice, okay, so say these little things are mice, the more it passes through the mice, the more disease it's going to cause. So sometimes, like with the flu might start out one way in like November, but then by April, because it's passed through person to person to person, it will be more virulent by the end of the flu season. Okay, now attenuation is how we can reduce pathogenicity. So there's different ways to attenuate something. So when we attenuate it, we're reducing the pathogenicity, and that's what we use to make vaccines. Okay, so we take a pathogen, and we modify it enough so that the immune system can have an immune response to it, but it doesn't make us sick. Okay, so that's the idea behind making vaccines and attenuating something. So you might take a virus and you modify it enough so that your body recognizes it as that virus, but it's not strong enough to make you sick. It's not virulent. Okay, so ways to achieve attenuation. One thing is taking like bacteria out of the animals and subculturing it on media. So if you grow it on media repeatedly, it loses its ability to cause disease. Okay, now a lot of things are done with molecular biology, so especially virus work. We can modify the genome, modify what the virus looks like so that it's not as pathogenic. It's not as easy. It sounds easy, but it's not as easy as it actually looks on paper. Okay, so now another way to do this is take the host and then we put it into a new species. Okay, and then we keep it from that new species and then we pass it through that new species over and over and over again. So the organism's going to adapt to the new host, and then we can use it to vaccinate against the original host. So it's lost its ability to infect this primary host. And that's how Pasteur developed the rabies vaccine. Okay, microflora. So this should be 10 to the 14th and 10 to the 13th. Okay, so there's a video here about the microbiome that's really interesting to watch. I think it's pretty simple, but it explains the microbiome really well. So microflora, microbiome, those are all the organisms that live on the body. They spent the past 10 years, the NIH has spent probably the past 10, maybe 12 years really starting to study that and trying to figure out the relationships between the bacteria, the fungi on your body and how they actually affect the health, how they affect different things. So we have 10 to the 14th microbes versus 10 to the 13th cells in our body. So we have more microbes in our body than we have our own cells. Okay, so there's a chart in the book where she talks about, she being the book author, about where the different organisms are. I don't think you have to memorize them or anything, but I don't want you to memorize where they are, what's where, but the idea is, okay, some places are colonized. Most places in your body are not sterile, but there are some sterile locations. So places like blood, the eyeball, cerebral spinal fluid, those are sterile. There shouldn't be anything in there, things that aren't exposed to the environment. There are some bacteria that are transient. They might go from one place to another, but they belong, you hear the dog groaning, they belong in your body. Your body needs them. They're really important to you. And they're trying to figure out more and more about how the relationship between the microbiome and your health. Okay, now opportunists are most of the time are organisms that take advantage of some normal function going wrong. So one example of a scenario where there might be an opportunist take effect or might take advantage is a host defense failure. So the classic example is if you have a wound in your skin. So your skin is one of your primary defenses. It really protects you from almost everything. But if you have a wound in it, it's much easier to get an infection. Okay, so sometimes we have microbes in unusual locations. So a good example of that is a urinary tract infection. And when little girls learn to wipe, okay, so there's E. coli in feces, but when they're learning to wipe, sometimes they get feces in the urinary tract, and then that causes E. coli's where it's not supposed to be, and it causes a urinary tract infection. We've talked about this before, I think. So microflora disturbances. So one, they also call these superinfections. So two examples are C. diff or Clostridium difficile and a yeast infection. So an example of a disturbance would be taking a broad spectrum antibiotic. And if you take the antibiotic, it kills all of the normal microflora and some organisms like C. diff and yeast. So yeast is a eukaryote, so the antibiotic isn't going to kill it. So then these will have a party, or they'll accumulate. Where C. diff has spores, and where it's located in your intestines, it's deep in the wall, so it doesn't get affected by the antibiotics. So then when all the normal microflora are gone, the C. diff will reproduce. Okay, so now we're back to Koch's postulates again. So this is the idea of trying to figure out what is actually causing a disease. So here's the dead animal, and we don't know what's causing him to die. So we'll take a culture from him, and we get a red, we get a purple, and we get a green. So we'll get these growing in pure culture, say they're bacteria, pure culture, and then we'll try to put them back into the animal. So that's step B, and try to see which one of them causes the same symptoms. So let's say we do get a dead rabbit, another dead rabbit, and it's from the one that we injected the red bacterium into, and then we can get the red bacterium out. So that whole series of things is called Koch's postulates. So the idea is we're proving what is causing the disorder. So if there's a new disease, Koch's postulates comes into play again. It's easy to do it with plants. We did it a lot with plants to try to figure out what was causing symptoms of different plants.